I can do that afterwards. I start with today's lectures and then the DNA methylation methods I will add afterwards. Okay.
Yes, sir. And by 5 o'clock, I plan for this valediction. Is it okay for you, sir?
When?
5 o'clock today, Indian time.
That would mean 1:30 here. Yeah. Okay. Oh, 5 o'clock. Let me look. So now, which time is 1:30 in 1:30 p.m. is now.
Yes.
In 4 hours. Okay. Yes.
Okay.
Okay, that's fine for me. I don't know whether today it will take me 4 hours, the lectures, but we can see.
Okay.
Maybe we have a little pause in between.
Yes.
Okay.
Yes.
Okay, so I start with the new methods, okay?
Yes, sir, please.
Okay. So can you see it?
Yes, sir, it's visible.
Okay, great.
Yes.
Okay, with this lecture, I would like to introduce some new methods in cancer biology, especially gene editing, some words on next generation sequencing, which is not really a very new method, but I can also show the next potential steps of evolution of next generation sequencing. I will speak shortly on single cell transcriptomics and 2 words on 3D cell culture. I am aware that there are many other new methods in cancer biology, but it is my feeling that these are the most important methods because many scientific work will rely on these methods. So it is important to know these methods, and for the decision of young researchers what to learn, in which field to become active, it might help to consider these methods, which will be very important in the next, let's say, 10 to 15 to 20 years. Okay, so let's start with gene editing. Gene editing, which had been performed before with other methods, which were much more difficult and much less efficient, has made a drastic change when the CRISPR technology was introduced. So where comes CRISPR from? CRISPR means clustered, regularly interspaced short palindromic repeats. Because that's, here I show you the prokaryotic cell, where the system comes from, because it's a kind of prokaryotic immunity against viral infection. And in the genome of many bacteria, you have a CRISPR locus. And the CRISPR locus contains Cas genes, that is a CRISPR-associated sequences. And then you have the clustered regularly interspaced short palindromic repeats. So you have repeats, the spacer and the repeat. So each repeat is followed by a spacer, and that's very regular. And the repeats are sequences derived from viruses. So they are integrated into the genome of the bacterium so that there's a memory of the virus that has been encountered, so bacteriophago. And this whole locus can be transcribed to form the pre-CRISPR RNA, where you have this typical stem loop structure of the RNA, followed by the spacer, the next RNA, next spacer RNA, and so forth. These are then cleaved to form single CRISPR RNAs, and these can recognize viral DNA in the cell and lead to the cleavage of the viral RNA. That's why it's a kind of immunity. So the viral RNA, once, if there's a memory for a virus, then the virus can be cleaved by this system. This was essentially identified and developed to its application in eukaryotic cells by the groups of Doudna and Charpentier, who won the Nobel Prize, if I remember well, last year or 2 years ago, I don't remember. For the first time, the Nobel Prize won by 2 women. So 2 women who shared a Nobel Prize. Okay. Let's say on the left side, you see how it works in the prokaryotic cell. You have, as I said, the CRISPR RNA that recognizes a specific site on the target DNA. And it has another part of the RNA that is recognized by the transactivating CRISPR RNA. Transactivating CRISPR RNA. And in the engineered version for eukaryotic cells, simply these 2 activities, CRISPR RNA and the transactivating CRISPR RNA, are contained in a single RNA that we call single guide RNA or simply guide RNA. Excuse me. The recognition of a specific sequence in the genome, so on the viral RNA in the bacterium or in the genome of eukaryotic cells, when there is a transactivating RNA, can be recognized by Cas9. And Cas9 has nucleolytic activity and introduces double-strand breaks. That's the whole process. So you get specificity through the CRISPR RNA that recognizes 20 nucleotides on the target DNA. So that's essentially highly specific because it's difficult to find 2 identical 20 nucleotide sequences in the genome. And the transactivating RNA allows for binding of the nuclease that introduces double-strand breaks. So what can you do with these activities? You can do a simply knockout. Why knockout? Because the Cas9 cuts and then the ends are joined by non-homologous end joining. And this is a very error-prone process. So often there is loss of some nucleotide or introduction of some nucleotide. And that leads to frame shifts if it's not 3. So if it's 1 or 2 nucleotides, you get a frame shift, and that means knockout of the protein, especially if you target the 5' part of the gene. But you can also introduce site-specific mutations. In this case, you need donor DNA, and we will immediately see how. And you can use Cas9 that introduces double-strand breaks, or engineered version, mutated version of Cas9, the Cas9D10A, that introduces only single-strand breaks. And in this case, you need 2 guide RNAs in order to cut both strands at slightly different positions. And in this case, the mechanism is no more non-homologous end joining, but homologous recombination. So the donor DNA that has large regions of homology and just contains a single nucleotide change, for example, binds to the region of interest and is integrated by homologous recombination after the cut of both strands. And that means also the knock-in. So if you knock in a whole gene, which is done by Cas9 with donor DNA by the same mechanism. The system has also been exploited by making fusions of the Cas9 protein in a way that the Cas9 loses its nucleolytic activity, but gains specific other activities. For example, the activity of methyltransferase or demethylase for DNA, or the function of transcriptional activators or repressors, so that the whole CRISPR system is only used to attract these enzymes to specific sites in the genome, in a way that you can methylate specific sites in the genome, or you can induce the activation or repression of specific genes in the genome. And similarly, you can also deaminate the cytosines in order to obtain mutations in specific positions, again with the fusion of Cas9 to cytosine deaminase. But let's see how it works in detail. So, site-specific sequence modifications. First of all, we speak about knockout. You have a gene of interest. Somewhere here you want to make a mutation, or you want to knock out this gene. The gene of interest, you design your CRISPR RNA and the transactivating CRISPR RNA. And in between of them, there is a PAM, a protospacer adjacent motif. So that's 4 nucleotides. And this whole thing is called gRNA. Normally, today, we have vectors where you just have the whole thing in it, and you just clone a nucleotide of 22 nucleotides. So the PAM and the CRISPR RNA, you clone it in this vector, so you obtain a transfectable vector that contains the whole guide RNA. You introduce that into the cell. The guide RNA attracts Cas9, and Cas9 makes a double-strand break. The double-strand break is repaired by non-homologous end joining, and in this process, often there is inserted or deleted one or several nucleotides. And then you have simply to detect whether you have obtained such events and clone to obtain the cell that carries these events. The efficacy of the whole system is relatively high, so that normally you should have a considerable proportion of your cells that actually carry the mutation. But it very much depends on the specific case. Not all guide RNAs work in the same way. Sometimes they do not work at all. So normally you would start with several gRNAs for the same gene in order to be sure that at least one of them actually introduces the mutation you wish. So if you have a single nucleotide substitution, so you want to introduce a mutation in a specific site. This obviously cannot be obtained by the process I described for knockout. You need another approach. Again, in this case, you use the mutated version of Cas9, the Cas9-D10A. You use 2 gRNAs that cut in different positions. And you design a donor DNA. The donor DNA is for the whole region identical to the target region, but it carries another nucleotide in the position where you want to substitute the nucleotide. So it contains what you want at the end, the sequence to be like. Cas9 introduces single-strand, Cas9-D10A introduces single-strand breaks, and they are repaired by homologous recombination repair in the presence of the donor DNA. So the donor DNA will be incorporated, and now you have the mutation in the position that you wished. This is somewhat less efficient than knockout, but still it has a good efficiency so that you can reasonably obtain mutated cells that carry a mutated gene. Much depends on the efficacy of transfection. So you should optimize your transfection protocol for the specific cell, because that is a rather limiting step in the whole process. And again, you have to screen for cells that contain, you have to clone the cells and then to screen which of the clones actually carries the mutation. But let's say if you come to an efficiency of about 20%, then 1 of 5 clones should have the mutated sequence. Obviously, there might be problems because the mutation could induce more or less viability. But that depends on the very specific case of mutation that you are introducing. For example, at the moment, we are introducing BRAF mutation in a normal melanocyte cell line, and we expect that BRAF the mutation gives a growth advantage, so it should be very easy to find them. In other cases, it might give a growth disadvantage, and then it is hard to find it. You can use the whole system also for insertion of a whole transgene. So if this is the locus of interest, you design again your gRNAs, and then you make a donor DNA that contains regions of homology to the outer sides of the external part of the locus, and in between you have the inserted sequence. So you will obtain insertion of the whole sequence. That can be relatively large sequences. So for example, entire genes, entire cDNAs. Okay, as to the fusion proteins, for example, here we have a fusion of Cas9 to a viral transactivator, WP64. And in addition, on the same plasmid, you have synergistic activation mediators and a marker protein, a fluorescent protein. And at the end, you have your gRNA. And you can target with the gRNA specific genes. And the gRNA will attract the Cas VP64 construct and activate transcription. And you can follow it by the activity of the fluorescent protein after transfection. And as you see, you get transactivation of genes that you have targeted. Not always it's very strong, but that might also be an advantage because in the contrast to cDNA overexpression, you get physiological activation. So if you use normal transgene approach, you get enormous overexpression of your gene, and then it's not so clear whether this is really what happens in reality. And here you get more physiological inductions. This is a method to introduce methylation in specific sites. You have the Cas9 fused to MQ1, which is a prokaryotic methyltransferase, and again with the fluorescent marker, the enhanced green fluorescent protein. And as you can see, this is the site of the target site. Untransfected controls have almost no methylation here. If you simply use Lac-Zeta, which has obviously no activity, still you have no activity. And if you use specific gRNAs, then you get an induction of methylation in this region, except for the target site of the gRNA itself, because that's covered by the gRNA. But all the rest of the locus is hypermethylated. There's some off-target effect if you use the same construct without the guide RNA. But that has been addressed. If you read the article, you see that there are other possibilities to avoid off-target effects. Similarly, another group has made a similar approach using demethylation with a TET enzyme fused to partially deleted Cas9, so a fusion protein between Cas9. The Cas9 obviously has lost the nucleolytic activity. It's fused to TET and led to specific sites in the genome where it can lead to demethylation. And similarly, if you make a fusion with the DNA methyltransferase, in this case, the eukaryotic enzyme, then you get methylation of unmethylated sites. And here you can see the results of specific demethylation or methylation of the locus. And at the end, as I told you, can also use CRISPR-Cas as a base editor if you fuse the Cas enzyme to cytidine deaminase, and it binds to the genome DA, and which is opened. The target is deaminated, and then DNA replication repair introduces a base-edited DNA. So instead of a cytosine, you introduce a thymine, and so you get a CT transition in a specific site. Obviously, you can target not only a single gene in your experiment, you can target many genes, or you can target all the genes. So you can transfect libraries of gRNAs, and there are libraries that contain 3 gRNAs designed for each gene. So it's about 75,000 gRNAs. And you transfect them into your cell culture. And you can expect that normally each cell receives a single gRNA construct. And then you can screen for the effects of the gRNAs. And you look at enrichment of specific gRNAs that obtain the effect that you are screening for. So in this case, the authors screened they screened for BRAF resistance. So essentially, they took a melanoma cell line, transfected 75,000 constructs of gRNA, and then treated the cells with vemurafenib, an inhibitor of BRAF, and expected that cells that had become resistant to vemurafenib have a growth advantage. And so they isolated the DNA from these cells that grew and sequenced the gRNAs contained in these cells. And you see a background of gRNAs, of all the gRNAs that you have transfected, but you see enrichment of specific gRNAs. So that simply means that you have sequenced more often these gRNAs than the others. So that's an enrichment. It's obviously not black and white, so you get an enrichment. But you can base your analysis on the p-value, on the probability that this observed enrichment has come about by chance, and then analyze a fair number of candidate genes for resistance to vemurafenib. And obviously you can apply this to any other aspect. So the important thing is that you have a good screening system. So you must have the possibility to distinguish cells that show the phenotype that you are screening for from those that do not show the phenotype. It must not necessarily be a lethal screening, so you not necessarily need something that kills the cell. You can also screen for some other aspect of the cell that you would like to do induce by transfection of a specific gRNA. But you need a good screening system so that at the end you can select your cells that show the phenotype that you wish, and then you can look in the cells which gRNAs are enriched in these cells. And the gRNAs obviously then hint at the gene that is normally important for the phenotype that you are analyzing. In this case, resistance to vemurafenib. And as I already told you in the lecture on CAR T cells, that's an example for introduction of a whole locus in a specific region. You have the local homology here that's contained in your construct, in this case an adenovirus-associated virus. You have the left arm of homology, the right arm of homology, and in between you put in a gene of your choice. So in this case, the CAR T, the chimeric antigen receptor. And you introduce it in a specific locus, which is selected by a specific gRNA. In this case, it's the T cell receptor locus. And then you get a chimeric T cell receptor locus containing the chimeric antigen receptor. This, as in this work has been shown, has the advantage of a more normal expression because it's in the correct locus, and the sustained expression of the T cell receptor, because in CAR T therapy, the problem can be that the chimeric antigen receptor is silenced after a while of activity, and then the disease can relapse. And this is prolonged expression of the chimeric antigen receptor if it's introduced into the T cell receptor locus. So what are we expecting to do at the clinical level? So there is a chance theoretically of in vivo gene editing. So you put your gRNA and the donor RNA in a vector and in a viral vector and introduce it into the patient. This is likely to work in situations where you need to correct the sequence in only a limited number of cells. For example, a monogenetic disease of the liver, where an enzyme is not produced. This has been done in the mouse, where the condition of the patient can much improve if a certain amount of cells in the liver contain the corrected sequence, because that's, for example, if the enzyme is not produced, after correction, the enzyme is produced, and even if it's produced by only a part of the cells, this might be sufficient to change the phenotype, or at least to ameliorate the disease condition, no? Because you have some enzyme produced. Much more likely is the application by ex vivo gene editing. Ex vivo means you take cells from the patient, modify the cells in the laboratory, and then you re-inject the cells. That can be T cells, very simply T cells that are modified and reinfused, for example, in the case of CAR T. But other applications are also possible. People have proposed to make knockout of PD-1 or CTLA-4, but that might be risky. You can use hematopoietic stem cells, modify them, or you can use any type of cell and induce the recurrence to stem cells and then edit them and re-inject them. The main limitation of this approach is to obtain the cells that you are really interested in. For example, we are studying in our laboratory, in addition to cancer, we have a project on the Friedreich ataxia. Friedreich ataxia is a disease characterized by a repeat expansion in the intron of a mitochondrial, of a gene whose product is important for mitochondrial function. And it leads to death of motor neurons in the cerebellum. And in this case, in order to do in vivo gene editing, you would have to reach the neuronal stem cells to modify the sequence in these neuronal stem cells. And that's very hard to achieve. And on the other hand, you cannot work with ex vivo gene editing because you cannot take off neurons from the patient, modify them, and re-inject them. So our approach is different. We try to screen with CRISPR libraries for genes that can compensate for the gene defect, in the hope that some of these genes might be drug targets, so that we can identify drugs that can improve the condition of patients carrying the mutation. So that's still relatively difficult to obtain. If you have heard on the experiments that have been done by a Chinese researcher who has modified a gene in pre-implantation embryos, this is much easier, but as you know, it's not considered to be ethical. Okay. Next generation sequencing. Just some words on next generation sequencing. I already told you that they rely on the possibility to parallelize the sequencing of single DNA molecules. And one of these approaches is emulsion PCR, where you introduce single DNA fragments in you bind single DNA fragments to an agarose bead and include the bead containing the sequence in a lipid sphere, in a lipid drop. So by making an emulsion in a way that each drop contains one bead, and then you amplify. This becomes a reaction container that contains polymerase and nucleotides, and then you can amplify the sequence on the bead, and you are pretty sure that each bead contains what has derived from a single DNA sequence. And then you can load the single beads on the sequencer to get the signal from each single sequence, or each single bead. Another approach, which I already showed you, is the approach by Illumina, the bridge PCR, where you bind DNA fragments to a solid surface by an adapter molecule. It makes a bridge, and when the bridge is formed, then it can be amplified in order that you get single locations on the array where you have identical sequences. So sequences derived from a single DNA molecule. And then you can sequence them by chemical sequencing and obtain the signal for each single sequence. And I also introduced to you pyrosequencing. That's the well chamber where you introduce a single bead, and then you do pyrosequencing, which generates a light signal that is proportional to the number of nucleotides incorporated. And each cycle you add just one nucleotide, and you measure whether it's included or it's not included. A modified version of this is the method applied by ion torrent, where instead of pyrosequencing, you just measure the release of protons by an iosensitive layer that transmits an electrical signal that can be measured. So you obtain the sequence in an electronic way. In many instances, you have to select the target that you wish to sequence. For example, it could be the whole exome. Then your target are all the exons of the exome. Or it can be targeted resequencing, let's say 500 cancer genes, what we call panel sequencing. And you have to select all these sequences, or you can also select the genes you are interested in, a low number of genes. And depending on what you are going to do, if you have very low numbers, it might be sensible to select your target by PCR. But if you have higher numbers of genes, then you would do hybridization, isolation of the target sequencings by hybridization, and that can occur on an array or in solution. And that's the main method that is applied today. There are other possibilities, molecular inversion probes or rainstorm technology, but essentially everybody today isolates the sequences by hybridization. Okay, once you have sequenced, the problem is analysis. So essentially, the development of the wet lab technology for next generation sequencing has led to a situation where it's relatively easy. So you have to make your libraries, you have to select your targets, and then you make your libraries, and then you just sequence. And that's not really difficult. But the analysis can be tough. Normally, you start with a base calling, a single analysis, base calling with a quality score, and you make an alignment to a reference sequence, in our case, the human genome. You map the reads to the reference and again score the quality, and then you can call the variance and visualize the variance and obtain a database of variation, and you interrogate databases of variation where you obtain a variant analysis. For example, you can filter for known genetic variants or for functional aspects of the variants you identify. Then you have candidates that you can validate and that eventually can be interpreted for clinical decision making in diagnosis or therapy. This can be cumbersome, and there are many workflow tools that are relatively simple, and especially Galaxy is a large effort to make next generation sequencing workflow that can be used by non-expert users. But in reality, I would strongly recommend to collaborate with a bioinformatician, and the bioinformatician most likely will work in the ambient of R R. There's an R project. Maybe I can show you. It's so let me. Okay. So that's the R project for statistical computing. And it's just r-project.org. That is free for everybody, and you have many functions that you can use for all kinds of bioinformatics. But it's a language, so you need to learn to compile in our language. But that's absolutely to recommend for if you want seriously start with next generation sequence analysis. This is a tutorial on next generation sequencing analysis resources. Here's the link to the website where you can obtain many informations on next generation sequencing analysis. Okay, but there are next generation sequencing is not the last step in sequencing evolution, although it's difficult to foresee how sequencing will really evolve in the next, let's say, 10 or 20 years. But one approach that is differing is smart sequencing. That means single molecule real-time sequencing, where you have essentially polymerase bound. You circularize your DNA molecules, which can be very long, and then they are sequenced by repeated cycles of synthesis in a well where just one molecule is contained. This method leads to highly accurate sequences and very long reads, and it's methylation sensitive because you get a light signal each time a nucleotide is incorporated, and when you have a methylated nucleotide, then the time interval is longer, so you can detect if the target DNA was methylated or not. Maybe I can show you a short video on this. Why doesn't it start? Okay, it should start. Okay, it doesn't work. Why doesn't it work? Yesterday it worked. Probably you can see it right now. I can change. But you can look after it again if you wish. But I just showed. Okay, that only for a short introduction of smart sequencing. This should not be. Oops, oh, excuse me, there's oh, excuse me. Okay. That's smart sequencing. And we have nanopore sequencing that again makes long reads. It doesn't use DNA polymerases and is also methylation sensitive. In this case, you have a protein complex that makes a channel, and the DNA is introduced into the channel. And when it passes in the channel, it releases a current. And the current is dependent on the specific nucleotide that is passing because it introduces different changes in the protein complex. And again, you can find here details on the technology. The real advantage is that this can be organized in a very, very small machine that is, let's say, smaller than a cell phone. And it's particularly interesting for field studies where you can carry on the device. You just connect the device to your computer and add the sample, and then sequencing occurs. Okay, that's so far on new technology, and now is a new approach that relies on classical next generation sequencing, which is single cell RNA sequencing or single cell transcriptomics. You have a tissue, for example, a tumor. You isolate single cells to be sequenced and obtain a signal for each gene in a single cell. So at the end, you have cell 1, cell 2, and gene 1, gene 2, gene 3 with the associated values. And you can compare gene expression in different cell types, cells and cell types. And then you can do bioinformatic analysis to identify cells with similar expression profiles, which form a population of cells. And eventually you can say there are known genes, that marker genes, that indicate to me that this, let's say, is a specific cell type, and this is another cell type that we know of. Normally, the data are presented by such a bidimensional, multidimensional reduction. So because it's a huge amount of data, but in order to get a grip on the data, you just present them in 2D and you place the single cell populations in the space according to their gene expression values. And this is done by UMAP plot, uniform manifold approximation and projection, which is kind of dimensionality reduction method. And then you have the different cell populations, and you can compare, like in this case, a control and a stimulated situation where you can see eventually differences in cell populations. An alternative to UMAP is T-distributed stochastic neighbor embedding, T-SNE, which delivers similar plots where you can identify populations or present in your sample. And obviously you can go then to analyze single genes and say for single genes in which population they are expressed. That is needed either to define the population, so to say this is the CD14 expressing population, and so it's a naive T cell or whatsoever. And this is the LIZ expressing population. And so it's identified in that way. On the other hand, you can also do the other way around, that you analyze in which cells once you have identified the cell populations, you can ask which cells are expressed which genes are expressed in specific cell populations. And that's a very potent approach. This has been used very recently in 2 articles that appeared in this month for mapping the developing human immune system across organs. And that's a very interesting study. I show here the B cell, where you have cycling B cells, mature B cells, immature B cells, separated by the UMAP plot, plasma B cells and B1 cells. So that are the different types of B cells. And then you can look in different tissues how these B cells are present in different tissues. That's shown here, where you get quantitative and that are the different types of tissues analyzed here. And here you get the quantitative distribution of single B cell types in different tissues. And that's a whole map for the human immune system across organs. And at the same time, there was a tabula sapiens, a multi-organ single-cell transcriptomic atlas of humans. Essentially, you have they isolated cells from 15 donors and did single-cell transcriptomics, including also the microbiome, in order to get a complex mapping of gene expression in different organs at the level of single-cell transcriptomics. That's the 4 donors that have contributed most, and here are the different tissues that have been used from these donors, and then the other 11 donors have contributed much less. So that's where this source of the data. And here, for example, you can see an example for T cells from different sources of the body, and where the clone ID of the different T cells and where they are located, where they go to. So this is the power of single-cell transcriptomics. And since in the course several times somebody asked on detection of early detection of cancer, this has also been addressed. And this paper, it's already 10 years old, shows that you can actually identify melanoma cells in the blood of a patient. You compare the single cell expression profiles to melanoma cell lines and to primary melanocytes and the circulating tumor cells. And you see that among the circulating tumor cells, you have similarity to melanoma cell lines, but not to primary melanocytes. So you can say these are likely to be melanoma cells that are circulating. And I told you yesterday that, or several times I have mentioned that, for example, the characteristic mutations, the driver mutations of melanoma, for example, BRAF mutation, is found also in healthy persons. And therefore, it cannot be used for screening, because you might find BRAF mutations in a healthy person and you don't need to treat him. So that doesn't mean that he or she has melanoma. But if you get an expression profile, then you are much sure about the malign nature of the cells, because it's not just one gene, but it's an expression profile similar to melanoma. But this article has not had so much follow-up, so it still is tricky. Still, probably it's not possible to do that. Okay. Another important development in cell biology is 3D cell culture. Why 3D cell culture? Well, okay, in our body, cells grow in 3 dimensions, and the normal situation of the Petri dish in the laboratory is not very representative of the normal growth of the cells in our body. So 3D cell culture has been introduced. That may be static suspension, so in a non-adhesive plate with a special medium, where the cells do not attach to the medium but grow as spheres. Another version of that is the hanging drop. So cells are actually growing in a hanging drop. This is especially and may form spheroids. This is particularly interesting because you can do that in high throughput, so you can have millions of hanging drops on your machine. That's a spinning flask, which was used, it's not more so much used, where cells are simply kept in solution. And you have a culture vessel that can be rotated, rolling bottles, magnetic levitation. You have today many microfluidic devices where you have channels where tumor spheroids can form, or you can embed the cells in gels, for example, agarose gels. And what you obtain are multicellular tumor spheroids. So because the real advantage is that you can include in the spheroid not only tumor cells, but for example, also fibroblasts, normal fibroblasts, or endothelial cells or perivascular cells, in order to obtain a more physiological admixture of tumor cells with extracellular matrix and non-tumoral cells that are normally present in the tumor stroma. And here you see an example of the difference between the 2-day monolayer and 3-day in a scaffold, that the cells really grow in a completely different way. It's more similar to tissues, and you get multicellular tumor spheroids. And that means an increased potential to derive primary cell cultures. So in 3D, many tumors grow in 3D, but not in 2D. So you get more primary cell cultures for more patients. And you can obtain a 3-dimensional tumor organization that is much more similar to the situation in vivo. Okay, I would make a break. Let's say 15 minutes. I need a coffee. Okay, then see you back in 15 minutes. Okay. Okay. Are you back? Perfect. So now I will, I can take questions in any case to the questions concerning the lecture on new methods. Are there questions?
There are no questions in the chat, sir, at the moment.
No questions. So, okay. You can think of questions in the meantime, and then we can discuss everything together at the end.
Yes, yes.
So now I will speak on the methylation analysis methods. Okay. Okay. So we had introduced DNA methylation as an important aspect of epigenetics. Normally, DNA methylation leads to repression of the gene expression, and sometimes it might be different, but normally methylation in the promoter determines silencing of the gene, and that's an important mechanism of gene silencing and regulation of gene expression. And it's in a way metastable. Let's say it's not as quick as transcriptional activation by transcription factors, but it's normally a relatively stable repression of genes. So it's used for the definition of the cell identity, and it's used in reprogramming and so on. So it's important to analyze DNA methylation. And the main approach to analysis of DNA methylation is bisulfite conversion of cytosine. So simply, if you add to cytosine bisulfite, you obtain cytosine sulfonate, which is then further transformed to uracil sulfonate and at the end to uracil. And as you well know, it is uracil is copied in replication, becomes a thymine. So you have essentially a cytosine to thymine substitution. And based on your reference sequence, you know where there are cytosines. And when you find a thymine instead of a cytosine, then you know this cytosine was not methylated, because 5-methylcytosine cannot be modified by bisulfite. So sequencing this one, you obtain the information that there's a cytosine. In this case, you obtain the information there's a thymine. So you can distinguish. Let me show that here. You have allele 1 that is methylated. Here is a cytosine that is methylated. Allele 2 is unmethylated, so it's the same cytosine is not methylated. All the cytosines are transformed into uracil, except the methylated one. If it's unmethylated, obviously also this is transformed into uracil. And then you do PCR, methylation specific or non-methylation specific PCR, and you can obtain the differentiation of bisulfite generated polymorphisms. That can be done with bisulfite specific primers, so that have a lower activity on them when there's a C, And normally you detect that by direct sequencing or pure sequencing, but there are many other methods also. But we use generally pyrosequencing, which is really straightforward, in order to see whether there's a C, which indicates it was methylated, or whether there's a T, which indicates it was not methylated. Methylating-specific primers are placed on the site of interest, where you get a reaction when there's a C, but you do not get a reaction when there's a U. As I told you, pure sequencing depends on a polymerase that incorporates a nucleotide, and by incorporating a nucleotide, it releases pyrophosphate. The pyrophosphate is substrate of the sulfurylase, which retransforms to ATP, and the ATP is a substrate for the luciferase, which releases a light signal. So each incorporation of a nucleotide releases at the end a light signal that can be measured. And you add one nucleotide at the time, you add a G and you get a G incorporation, then you get a C incorporation, T is not present, you get an A incorporation, and here you have two Gs because simply both of them are incorporated and they just yield a double intense light signal, and then CC and T and so on. That's pyrosequencing. And obviously you can do that on microarrays. You can also analyze methylation on microarrays, specific for bisulfite treated DNA. And you can do it obviously by next generation sequencing. Again, you have bisulfite conversion, the first step, where you substitute an unmethylated C with a U, and the methylated C remains unaffected. You do PCR and fill in of the ends, adapt ligate adapters, and then you do size selection and quantitation, next generation sequencing, and you get a profile for bisulfite treated and non-treated, and you can compare normal to cancer in order to understand methylation, differential methylation. A special application of this is the reduced representation bisulfite sequencing, which is today the main method used for analysis of DNA methylation by next generation sequencing. In this case, you do a restriction enzyme digestion of your DNA, do fill-in reaction and repair, and a tailing, adapter ligation, size selection. Then you do the bisulfite conversion, PCR amplification, and sequencing. Which is the advantages that you pre-select the sequences to be sequenced by a restriction enzyme digestion, and you use restriction enzymes that are typical, that recognize CpG islands. So that you have a reduced representation because it's mainly based on CpG islands. Smart sequencing, as I showed you before, also can analyze DNA methylation because the incorporation, without any modification, in this case, you do not need bisulfite conversion because the incorporation of a T as opposed to a methylated C takes longer time than other incorporations. So you can decide from the nucleotide incorporation kinetics measured in real time whether a nucleotide is methylated or not. Okay, that was very short. But that are essentially the methods for analysis of DNA methylation. So essentially, if you have just one gene that you wish to analyze, then I would recommend bisulfite conversion and pyrosequencing. That's at least what we are doing here. If you have, let's say, 10 genes, still you can do it in that way. If you have many genes or the whole genome, then you would use reduced representation bisulfite sequencing. And obviously you can also do next generation sequencing with a panel of genes that are modified by bisulfite sequencing. By bisulfite treatment, excuse me. Okay. Are there any questions to this talk or to the talk before? Today we have also children in the audience.
I just muted them, sir. Any questions from the participants? I think there's nothing at the moment, sir.
Okay. So whenever you have questions, don't hesitate to make the questions. Okay, then I would go further with the next talk on. The next talk is on breast cancer risk in India, and that was very interesting for me because I had to look in the literature what is known on breast cancer in India. I did that before, and I have done it again. And so it's new information for me, and maybe you can add something from your own perspective. You are living in India and you know the situation. I'm only based on the literature, but there are very interesting aspects, and especially as you will see in the talk, in India there are screening programs being implemented, and that's a huge step forward, obviously, but it has also drawbacks, and that should be considered. And yes, okay, let me start. Okay, no, that's not. That's the last slide. So breast cancer risk in India. Breast and cervical cancers are the two most common cancers among Indian women, and they contributed almost 40% to the total cancer burden in India in the year 2020. And why are we speaking on breast and cervical cancers? As you will see in the talk, they have opposed tendencies. Breast cancer is increasing and cervical cancer is diminishing, but they have a high prevalence. Breast cancer has high prevalence in affluent countries. Cervical cancers have high prevalence in less affluent countries. And that's what probably explains the change that is occurring in India. India has a population-based cancer registries, and so on, more than one. So it has population-based cancer registries that were established since '81 under the National Cancer Registry Program, National Center for Disease Informatics and Research of the Indian Council of Medical Research in Bengaluru. So that's a very advanced registry program that allows for epidemiologic studies of diseases, and that's a very good achievement. So according to the recently released NCRP 2020 report, breast cancer age-adjusted incidence rates for 100,000 of population was high in metropolitan and urban cities, ranging from 48 in Hyderabad to 7 in Meghalaya. The age-adjusted incidence rates of cervical cancer was high in the northeast region, followed by Bangalore and Bashi, rural regions of India. It ranged from 27.7 in Papumpare district to 4.8 in Dibrugarh. So you see already that there is a difference between metropolitan areas, urban areas, and rural areas. And that's the data, numbers and age-adjusted incidence rates. And what I would like you to focus on are the estimated annual % change for breast and cervical cancer. So that means you have in Bangalore, you have an increase of 2.7% per year of breast cancer. And that's similar, that's lower in rural areas, but still it's an increase. And Chennai, which is more important for you, you still have a high increase. So 1%, 2%, 3%, that might not sound much, but it's per year. So it's really dramatic increase. On the other hand, you have a dramatic decrease in cervical cancer, almost everywhere. And here you see the curves, where you can see that in all these regions, you have an increasing number of breast cancers and a decreasing number of cervical cancer. And it has, breast cancer has substituted cervical cancers at the first position. So breast cancer today is the cancer with the highest incidence in Indian women, except for Bashi, which is, as I understand, a rural region where the crossover has not yet taken place. But you can, that was 2012, probably by now. Breast cancer is also the most prevalent cancer, and also in Bashi. That's the rate per 100,000, age dependent. And you clearly see that it increases over time. But you also see that there is a steep increase early on. So relatively young women. It's a little less clear for Bashi, but for all the other regions, it's a similar tendency. Breast cancer incidence is increasing and cases augment very early in life. So in cervical cancer, on the other hand, you see a decline from 1987 to 2012 in almost all regions, a little less pronounced than Bashi, but you see that in all the areas. And here's just another representation where you can better see the age dependency. And you have a very steep increase from in very early years in life in all the regions. And that's a little specific for India. The risk factors for cervical cancer are lower socioeconomic status, HPV, human papillomavirus prevalence, earlier age at marriage and first intercourse, multiparity and tobacco consumption. As per the National Family Health Survey, a higher proportion, 9.2% of women aged 15 to 19 years who were already mothers or pregnant during 15-16 were seen in rural India compared to urban India. It's almost the double. In rural India, women use any kind of tobacco, 8.1% compared to urban India, half of it, 4.4. And for the total fertility rate in India, reduced from 3.4 per thousand in 92-93 to 2.2 in 15-16. So parity is lower now. And the breast cancer risk factors include higher socioeconomic status, early ages at menarche, later age at first birth, lower parity, obesity, lack of breastfeeding, urbanization. These are linked because lower parity, later age at first birth is more typical for urban areas than for rural areas, but they are not necessarily independent risk factors. The NFHS data showed that women who are overweight or obese, so that means a body mass index of over 25 kilogram per square meter, has increased in urban India recently, 31.3%, compared to previous survey from 2005 and 6, where it was 28.9. Though obese women also increase in rural India, the percentage is less than half, 15% of urban during NFHS 15-16. Children under age of 6 months that were exclusively breastfed was high in rural compared to urban India. This is not such a great difference, 55.9 to 52.1%, but still it's higher. The national level changes in risk factors were underlined by the NFHS. However, the changes in the risk factors may vary between the registry area and the population. Another particularity of Indian breast cancer is the ratio of triple negative breast cancer. As you may know, triple negative breast cancer is particularly aggressive and is the hardest to be treated, and often, unfortunately, leads to the death of the patient through metastasis. And if you compare the percent of triple negative breast cancer, triple negative means no expression of estrogen receptor, no expression of progesterone receptor, no expression of HER2. So the three estrogen receptor and progesterone receptor, which are typical for hormone receptor positive breast cancer, HER2, which is a class of its own, and for which there is a drug, trastuzumab, that is quite active. And triple negative breast cancer expresses none of these receptors, and therefore it's much harder to treat them. And essentially, we have only chemotherapy, but chemotherapy only a part of these cancers respond to chemotherapy. So it's particularly hard if you have a high ratio of triple negative breast cancer. And if you compare India to Western countries, USA, here's Italy, Germany, no, then you see that it's much higher. It's more than double what you observe in Europe, let's say Germany, London, Italy, no, Netherlands, it's even lower. And this is really concerning. You see that it might be divided by other countries in Asia. Oh, that's Algeria, it's North Africa, but in Indonesia and Pakistan. But still, it's quite typical for India. And there are differences in the regions or different cities of India. and you see Chennai is 25%. But you go up to over 40% in Nagpur. The prevalence of triple negative breast cancer here shown by various study characteristics. So that means you have studies from east, north, south, and west. And there are differences essentially between north and south, but that's probably in some way linked to the socioeconomic status. As I understand it, south is richer than the north in India. There's not so much difference between east and west. The study size, that are, let's say, quality measures of the study, because if you have many patients, the result is more reliable. And you see that you are always around 30% of prevalence of triple negative breast cancer versus all breast cancers. And it's more reliable if you have a larger study. And essentially you see that the confidence interval is smaller. Less participants, you get more varying results. No? And that's how estrogen receptor status was defined. If over 1% of estrogen receptor positive cells were considered positive, if this was not the case, then you have a larger confidence interval. If it was the case, then you have a more reliable result, and even worse if it was not reported, because nobody knows how they did the analysis. Similarly, HER2, which is internationally classified in 3+, 2+, or negative, where 3+ is considered positive, 2+ is normally unclear, and in many cases cleared then by in situ hybridization, and one below 2+, it's considered negative. Again, if this is stated, then if this is applied, then you have a more reliable results. And so the whole study quality, if the study quality is high, then you get smaller confidence intervals. You actually get also a smaller prevalence than by lower quality studies. But what we can rely on are the high quality studies. You see that. So we are pretty sure about the prevalence. So it's not an artifact from the studies. That's the projected incidence of cancer statistics in India 2020 for male, female, and both sexes. You see that the cumulative risk in 2020 for male is 1 in 9, as it is for female. For breast cancer, obviously, in men, it's much lower. There is male breast cancer, but it's rare. And you have 1 in 29 in female, so you have over 200,000 cases expected in 2020, 205,424. But as I understand, the National Health Mission is actively working on this problem by health promotion through behavior change with involvement of community, the civil society, community-based organizations, and the media. Outreach camps are envisaged for opportunistic screening at all levels. So screening is introduced in the health care delivery system from subcenter and above for early detection of diabetes, hypertension, and common cancers. So obviously the prevalence of diseases is not limited to breast cancer, but also diabetes and hypertension. Management of chronic non-communicable diseases, especially cancer, diabetes, cardiovascular disease, and stroke, through early diagnosis, treatment, and follow-up, through setting up of NCD clinics. So new clinics are implemented to build capacity at various levels of healthcare for prevention, early diagnosis, treatment, operational research, and rehabilitation, provide support for diagnosis and cost-effective treatment at primary, secondary, and tertiary levels of healthcare, and provide support for development of database of non-communicable diseases through a robust surveillance system to monitor NCD morbidity, mortality, and risk factors. So that's a very ambitious program, but already during 15-16, more than, I guess, I have calculated 1.29 crore is 12.9 million persons have been screened in the designated NCD clinics. So that's many patients, many people, healthy people that are screened. Among these NCD clinic attendees, around 8% were diagnosed to be diabetics and 12% were hypertensives. So two conditions where an appropriate intervention can change completely the prognosis. So diabetes, it's obvious, but also hypertension. Also, around 90,000 persons were diagnosed to be suffering from cardiovascular diseases, and over 13,000 persons were detected to be having common cancers, including oral, cervical, and breast cancers among them. During 15-16, about 96 lakh, that's 9.6 million, I guess, persons were screened under various outreach activities for common NCDs in different facilities, and they were referred to higher centers for diagnosis and management. So there is being done something, it's going on. For the cancer component, there's a tertiary care cancer center scheme, which aims at setting up strengthening of 20 state cancer institutes and 50 TCCCs for providing comprehensive cancer care in the country. Under the scheme, there is provision for giving a one-time grant of a certain amount of money for the state cancer institutes and the TCCs to be used for building construction and procurement of equipment, with the center to state share in a ratio of 60-40, except for the northeastern and hilly states where the share is 90-10. So that's actually a program, a very sensible program to improve the coverage of prevention and diagnosis and treatment for cancers and other non-communicable diseases. But if you start doing mammographic screening, then you have to expect overdiagnosis. And this has been much discussed in Europe and in the United States, and still the issue is not settled, because you get different studies, different types of studies that come up with different estimates of breast cancer overdiagnosis, but you see numbers, well, very different numbers, so we aren't sure about that, but they are relevant numbers. And it's a similar estimation shown as ratio of the number of breast cancer deaths averted to cases overdiagnosed by mammographic screening. So for one case that has a breast cancer and dies from it, 3 cases are diagnosed as breast cancers, but they are not really breast cancers, or at least they do not put the life at risk. And that's, again, very much changing in different studies, but there is some overdiagnosis in any case. So you should expect, by introducing what epidemiologically this leads to a high number of new cases. So it might determine an additional increase in breast cancer incidence, because you identify many situations as breast cancer that normally would never have been detected. That's especially true for ductal carcinoma in situ. Most ductal carcinomas in situ never develop into a cancer, but they are detected by mammographic screening. If you don't do mammographic screening, they are not detected, and they do not develop into cancer, so they are never detected. If you do mammographic screening, you detect many ductal carcinoma in situ, which by any definition is a carcinoma, but it has no chance of, it has a very low risk of developing into aggressive cancer that can put the life at risk. So you get an over incidence of breast cancer if you do mammographic screening. And that is, you can see that in the data from Europe or the United States, where in the 90s, you had a sharp increase in the breast cancer incidence because mammography was introduced. But mortality did not change much. And that means you detected many cancers that are not really threatening anybody. It's even worse with melanoma, because melanoma awareness has much increased, and melanoma screening has been introduced. And so many melanomas that would never develop into a full-blown melanoma are detected and classified as melanoma. So you have an enormous increase in incidence. In melanoma, actually, you have a decrease in mortality, despite an enormous increase in incidence. That means, on the one hand, you detect many melanomas that are not really dangerous, and on the other hand, you see the effects of immunotherapy, that mortality has declined a lot. Okay, I would be very happy if you could comment on the cancer risk in India, what is going on in India, what the government is doing, if that is enough, if that is perceived by the population. I would be very interested to hear your opinion. And I see there are questions. What are the target proteins to concentrate on in lung cancer? That's a tough question. I mean, lung cancer is one of the cancers that best responds to immunotherapy. So we should, and we are actually focusing on immune-related proteins in lung cancer, so PD-1 expression, PD-L1 expression, for example, also the soluble form of PD-L1, which is a marker for response to immunotherapy. If you intend proteins that are at the base of the tumor evolution, you know that there are different classes in lung cancer, non-small cell lung cancer. Especially, you have frequent mutations of the EGFR. And that's important because I don't know the data from India, but I know that there's a difference between Europe and Asia, at least Japan, because in Europe, EGFR mutated lung cancers are about a small proportion, less than 10%. Whereas they are pretty prevalent in Japan, and I don't know in India. And that's important because EGFR mutated cancers respond to gefitinib, an inhibitor of EGFR signaling. But I mean, there are many other proteins to target, to look at. Other type of non-small cell lung cancer normally carries a mutation in the ALK gene, for which there's also some kind of therapy. And I think many others are to be detected. But I think at the moment, the main, also looking at our experience with other cancers, targeted therapy normally gives a good response early on, but after a while, let's say after 6 months or a year, the cancer becomes resistant to targeted therapy. And so it's only a transitory effect, whereas immunotherapy has a long-lasting effect. Maybe not immediately, but then you see long-lasting effects, at least on a portion of the patients. So that's because I would, that's why I would recommend to study in more detail the response to immunotherapy also in lung cancer. What is the effective phytoconstituents against lung cancer? I'm not really aware of phytopharmaca that have real effect against lung cancer. I always see in phytopharmaca and phytoconstituents more a chance for tumor prevention. Essentially, the prevention of lung cancer, the first step in prevention is smoking cessation. People should not smoke. That would lower cancers a whole lot. But still, even people who do not smoke can develop lung cancer. So there's another group that is in some way linked to pollution in urban and industrial areas, which should be addressed and which probably will be addressed because all of the world is speaking about substituting fossil fuels with renewable energy. And I know that India has a program of leaving carbon usage for energy production, and that will surely improve also the air quality and lower the lung cancer rate among non-smokers. But again, I'm not aware of specifically of phytoconstituents specifically aimed at lung cancer. In my own experience, I work with curcumin. I would recommend curcumin anyway because it probably lowers inflammation and therefore can lower the risk of cancer development. Do cosmetics play a vital role in cancer for women? I would say no, not that I would know of. Provided that you use controlled cosmetics that do not contain toxic compounds. But I think that's normally today, cosmetics do not contain toxic compounds. So we should use cosmetic to children or not, cream or powder. I have 3 children, which now are grown up, and I used obviously a little cream and powder because once for the time that they wear the Pampers, or they call if they are not continent, they need some cream and powder. I would guess in my own experience. No, so I would say I would recommend to use as less as possible. But there are also, I think that there are products that are very, very controlled and very good studied. So I wouldn't see there a major problem. I guess that if you want to do something for your children, look at the diet. I don't know in India, but in Europe and in the USA, many children are overweight or obese, and especially if they start to be at childhood, then they have really an increased cancer risk. because the whole metabolism gets used to an excess of sugar or carbohydrates. Another point is try to avoid meat, no, because to eat less meat, no, that should be easy in India, because traditionally the Indian kitchen is not so heavily based on meat like European and Western diet. No, that's much more important, I think. Cosmetics, I don't think it's a real problem. Study material, but I haven't touched study material you would recommend to research or study on lung cancer. So I actually, in the whole talks, I haven't touched so much on lung cancer. But I would, well, you will receive the study material, which is essentially consists in all the articles that I have cited in my different lectures. There's not so much on lung cancer. But if you wish, I can send also a small collection of papers on lung cancer that you could study. I have to take note that I must think of lung cancer. Okay, if you can go on. Yes, I can send. You can go on thinking of questions. I would like to drop 2 words on the questionnaire, because we have prepared a questionnaire for the whole series of lectures, and there is essentially, to each lecture there are several questions. It's a multiple choice test. You have 1 question and 4 answers, 4 potential answers. Only 1 is correct. There's always only 1 is correct. And I recommend to read very carefully the question, because sometimes the question is, which is not the case, no? And so you see 3 answers that are clearly the case and 1 that is not the case, and then you should make your sign on the answer that is not the correct answer. And yes, what else can I
tell you? Sir, I want to ask 1 question.
Yes.
Sir, or is there any chances of cancer to use of soap?
Cancer for users of?
Soap. There are different types of the soaps are available in the market. This is also cosmetic product.
I don't think that there are really data on it. I think soap is not soap. You have natural soap, and you have soap containing chemistry in it. And I would recommend to stick to natural soap. And I don't see, I don't think, and I know that there are no studies on cancer risk of soap. But it should be careful. It should be recommendable to use natural soap instead of paraben containing soap and so on.
Because I have read in some papers there was some home cleaning agents are also very harmful.
Yes. Yes, home cleaning is okay. If detergents, it's worse. I thought of soap for your body, but if you go to house cleaning agents, you can have also pretty harmful agents. Although I don't think that there are studies. I don't know if you have read articles. That's interesting. But again, there are alternatives. No, it's also for the ambient For the environment, it's much better if you use detergents that are fully degradable, that are biologically degradable. For example, in many instances, just acetic acid is sufficient to clean many surfaces, and you don't need heavy chemistry. No, that should be a trend in a world where we become finally more aware of what we are doing to the environment. So use natural compounds, use biodegradable detergents. Definitely.
Sir, yes, sir. You was telling about the Indian context. Cancer problem of in women is my very interesting area. So, sir, we know the uterus cancer is very dangerous and it is known as a silent killer. Why we cannot identify in the earlier stage or first stage?
Well, at the moment, the earliest stage where you can identify it is by mammographic screening, and you normally detect lesions that are some millimeters in dimension. That sounds small, but it's already many cells. So there's definitely an advantage in survival for mammography screening, because you take them out earlier, and that means they have less time to disseminate, and you can start treatment earlier. But still, it's let's say a tumor of some millimeters. It's millions of cells, and it would be much better to detect it even earlier. But that's at the moment it is not possible. I showed you the attempts to do it by circulating tumor cell screening, but still this is not a measure for cancer diagnosis. It's not ripe. It's not been developed enough to do cancer screenings. And I don't know whether it will ever be possible. We use circulating tumor cells for the detection of minimal residual disease. So once you have detected the cancer and operated the cancer, then you can check in the bloodstream whether there are still cancer cells that indicate that there is some cancer left or that there is metastasis developing. And that means also that you can make early detection of relapses. That's possible. But screening by circular tumor cells, likely also too expensive to do it on the whole population. We have other two questions. Yes, the processed soy-based products, which nowadays substitute non-vegan, like vegetarian meat, veggie chicken, is healthy. I'm convinced they are healthy. I haven't ever heard of problems associated. It obviously depends on how the soya is cultivated. If the soya is cultivated with the heavy use of, but it's not so different from, it's definitely healthier than meat, because the animals also eat soya, and they would accumulate pesticides and so on that are used in the cultivation of soya. But we should come more and more to less usage of pesticides. And so I would say at the moment already soy-based products are healthy. Also, how impactful is mobile phone uses and its radiation cancer prevalence? Okay, let's say if mobile phone would have a real impact on cancer, then we would have seen it. Because in early days of mobile phone usage, the radiation of your mobile phone was much heavier than it is today. And now it's almost 30 years that there are mobile phones around, and we would have mobile phone dependent cancers. And I think we would have seen it by an increase. Normally you would expect brain cancers or let's say testicular cancer, something like that, or because you keep the phone in your pocket, and we do not see it. And today, the exposure, radiation exposure, is much reduced in comparison to the early days of mobile phone. And also the radiation, the highest radiation you can receive, you receive from your mobile phone, not from the 5G masks, 5G plans, no? So it's actually, but I don't think that there's a, at the end, it's always like that. If you consider a world without mobile phones and a world with mobile phones, I'm sure there will be cases, because some people have some kind of genetic predisposition to brain cancer, which normally yields a certain rate of brain cancer. And if stimulated by radiation from the mobile phone, they just get some more cancer. But it's not relevant numbers. Otherwise, we would have seen. Okay. No more questions?
There are no questions at the moment.
Will the questionnaire will be done immediately or after a while?
No, sir. Today evening, I have scheduled it for 8 p.m., 8 to 9.
Okay. Okay.
So after valediction, they will have a short break and then they will start it.
Okay. So I hope it's not too difficult. I try to be basic, but still it's a lot of information in the course.
Yes. Yes.
And well, is there a threshold they must receive?
Definitely,
Yeah.
sir.
Yeah, okay. I hope it's not too high.
Yes. Yes.
Okay.
Okay, sir.
So we see back in
Yes. Yes, sir.
40 minutes.
Fine, sir. Okay. 5 o'clock.
5 o'clock Indian Standard Time.
Yes.
Okay.
Yes. Yes.
Okay. Thank you. And everybody will be there. Also
Yes, including
all the
participants. Yes. all
students. So I can thank everybody at the end.
Yes.
Okay.
Yes.
Thank you.
Thank you.
See you later.
Yeah. Fine.
Selvamani.
Sir, good evening.
Dr.
Sir.
Selvamani.
Good evening.
Start பண்ணலாம் sir.
Start.
Join. Okay.
Okay.
Sir.
Professor also joined, sir.
Kumar sir. Good morning.
Sir, good evening, sir.
Good evening.
Sir, good evening, sir.
Good evening.
So, good evening, one and all. It's very pleasure for me to invite you all for the MHRD sponsored GIAN virtual course on new concepts in tumor biology. So we have 5 days intensive lecture on various topics held by Professor Ulrich Pfeffer from San Martino, Italy, who is the head in the tumor epigenetics unit there. And he was very much supportive for me since from the application time and in the organization of course schedules, handling the subjects, conducting the exams. So I welcome you, sir. And Professor J. Kumar, Honorable Vice Chancellor of Madurai Kamaraj University, will be the chief guest today, who has graciously accepted my invitation. And he is very much interested in our course since beginning, since during the application time as a Director of Center for Technology Development and Transfer at Anna University, Chennai. And our Dean, Professor Senthil Kumar, who is always supportive to me in all my academic activities and research activities. So I welcome, and I would like to introduce Professor Tiruchirappalli, Head of the Department, and Professor Rukmani, Director, Center for Excellence in Nanobiotranslational Research, for this valedictory event. So formally, let me start the program, sir, please, with all your permission.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
Whether my screen is visible, sir?
Yes, your screen is visible. Yeah.
Yes. Let me invite Professor T. Senthil Kumar to welcome the invitees and audience.
Good evening to everyone present here. Myself, Dr. T. Senthil Kumar, Dean, University College of Engineering, and University Tiruchirappalli. Honored to welcome you all for this validity function on MHRD sponsor GIAN Global Innovative for Academic Network virtual program on new concept in tumor biology from bench to bedside, conducted by Department of Pharmaceutical Technology, University College of Engineering, and University Tiruchirappalli. I'm indeed happy to welcome you all eminent personalities who are presented for this validity session of this FT. First and foremost, I would like to extend my warm welcome to Honorable Vice Chancellor of Madurai Kamaraj University, Professor Dr. J. Kumar, and also our previous Registrar of Nadu, Chennai, who had supported us on all our endeavors in our college development. To mention a few, Dr. J. Kumar is an instrumental tool for construction of AICT funded student hostel with worth of 2.5 crores at BIT campus. Also with the Professor entire help, construction work of the compound wall completed in protected 354 acres of land belonging to university. In spite of many administrative and legal issues, the compound wall perimeter is 9.4 kilometers covered in the full entire university. I take this opportunity to thank our Honorable Vice Chancellor of Madurai Kamaraj University, Professor Dr. J. Kumar, also our previous Registrar of Anna University, for his support and cooperation for this forum. In this session, on behalf of University campus, and then as a own, as well as faculty members and my student friends, on behalf of everyone, I welcome you, sir, for this validity session. Welcome you, sir. I take pleasure to welcoming course faculty, Professor Ulrich, Head Tumor Epigenetics, Scientific Director, San Martino, Italy, who has with us. Thank you for welcome you, sir. I am happy to welcome our own team from Anna University, Professor Dr. A. Prochikudi, Professor Head, Department of Pharmaceutical Technology, Dr. Professor K. Rukmani, Professor and Director, Center for Excellence in Nanobio Translation Research, and Professor Selvamani, Professor in Department of Biotechnology, Pharmaceutical Technology, for coordinator for this GIAN course, and other head of the departments, faculty members who are present here. At this juncture, I would like to appreciate Professor Kumar for accept our invitation to validate for this section. Also, I thank Selvamani for coordinating this event. And this also, I'm happy to welcome all the participants of various institutions for attending in the online course. I hope this course will enrich your knowledge and opening a research perspective in our area of tumor biology. Welcome you again, the validity function. Thank you once again for the opportunity.
Thank you so much, sir. So let me invite few participants to volunteer yourself to give a feedback about this course and other things.
Good evening to everyone. This is Surya, doing my second year BTech Biotechnology degree at PSP Engineering College, Karur. It has been such an honor to be a part of a wonderful event. First of all, thank you for Professor Ulrich for his amazing lecture. And thank you for organizing committee for arranging these kinds of events, which would be helpful for our future studies and to work in our major projects. Through this course, I have gotten a depth knowledges in tumor biology, breast cancers, and their treatment methods, and lots of pathways and mechanisms. And I am requesting our organizing committee to arrange more courses like this in various streams. Thank you.
Thank you, Surya, for your feedback.
And thank you, sir, for arranging such a great course.
Thank you so
and
much.
Organizing it. Thank you.
Good evening to everyone. My name is Sharmini Tyagaraja. Actually, I'm from Malaysia, but residing in Chennai right now. So I took this opportunity to enroll myself in this course. I find this course is very much eye-opening and bring me further advance to learn more on epigenetics especially, as well as a new method in cancer biology, because I also impart my knowledge in biotech and also stem cell. So with this course, I find it further information, which I have to be very thankful to Professor Ulrich and also to Dr.Selvamani, who has extended his help for me to enroll into this course, so that and also made this possible for me to learn. Because we don't find this kind of a course back in my hometown in this extensive manner. Plus, the notes and also the entrenched sharing has given me further knowledge to expand my research as well in this field. Plus, I find that the course outline as well, the content and the understanding, this is just my point of view. If I would, probably it's my way to understand, which I prefer on an animation scale, which I find it during Prof. Ulrich towards the end, he shared the which is more I find it more understandable, easier. So compared to the notes that we conventionally share, but sorry, but this is just my thought. But I'm seriously looking forward for this again, for a better opportunity and explore new findings in this era. Thank you, Prof. Ulrich, and also thank you, Prof. Selvamani, to give this opportunity.
Thank you so much for your words.
Thank you.
Anybody else?
Pleasant.
I am Krishna, business scholar from NCTBBB. I found this meeting very useful and informative, and each sessions are very well organized. And I thank everyone working together for such something online. And I also thank Dr. Ulrich and for sharing his years of knowledge and insight and advancement of biology. And I also like to thank Dr. Selvamani sir and GIAN for giving us this opportunity to hear from MHRD. Thank you.
Thank you so much.
Thank you.
Anybody else would like to share your feedback?
Pleasant evening, everyone. This is Soumya J, Master of Technology, Department of Pharmaceutical Technology, University College of Engineering, Tiruchirappalli. First of all, I would like to thank and extend my sincere gratitude to Professor Ulrich for the immense lectures. I would take this opportunity to thank each and everyone working behind the curtains for the successful completion of this program. This gave opportunity for students like us to get a vision on what tumor is, starting with a simple definition to advanced concepts in the tumor biology. The presentation slides would also be definitely helpful in the future. I would like to thank Professor Selvamani for coordinating such an informative course and giving all of us an opportunity to participate in it. Thank you all.
Thank you so much.
Thank you so much. Anybody else would like to share your words? Next, let me invite Professor J. Kumar. Previously, Professor Kumar, at the moment, he is Honorable Vice Chancellor of Madurai Kamaraj University. He is having a teaching experience of more than 29 years with a research experience also. He was previously serving as a BSR faculty fellow at the Crystal Growth Center of Anna University, and he has completed more than 20 sponsored research projects and has 2 patents to his credit. He published more than 160 papers in peer-reviewed international journals and has guided 25 research scholars for their PhD. As our dean told, he already served as a registrar of Anna University and other administrative positions, including the director of Center for Technology Development and Transfer, Planning and Development at Anna University. He organized many international events, and he was very much instrumental in implementing many administrative reforms in the Anna University. He is the recipient of Best Innovation Award, DSSTM from the University of Parma, Italy, and Tamil Nadu Scientist Award for the year 2012, and the Department of Atomic Energy Young Scientist Award for his contribution to his physics. Mr. Kumar has conducted several collaborative research and teaching in foreign institutions and visited 15 foreign countries for academic and research purposes, and he is proficient in German language also. So let me invite the respected Vice Chancellor of Madurai Kamaraj University to give his chief guest address, please. Please, sir.
Good evening to all our Indian colleagues and good afternoon to my friend from Italy. I hope he had his lunch. Professor Ulrich, you had your lunch?
Yes, I have.
Okay.
Thank you. Thank you.
I hope you didn't stop your lunch.
No, I had time enough. Thank you.
Okay. So thanks to Dr.P.Selvamani for initiating this program. We started this course or the GIAN program way back in 2015. And so for 7 years, it has been going on successfully. And a lot of efforts had gone behind the organization of this program. And you must be knowing by now, it is not a program which is just funded and then you invite a friend or an expert and then go on with the program. It goes through a rigorous process of evaluation, and then you have to organize the program with the right kind of participants, and they have to also contribute by way of understanding the lectures and then kind of experiencing an evaluation at the end of the program. So this is a very important program, and to summarize this program, for many of you who may not have known as to how it all started, the IIT Kharagpur in India initiated a program to interact with an expert from Japan to formally evolve an activity in India to make progress in fast or super fast trains. In Japan, as many of you know, it's called as the conventionally an English term, it's called as bullet train, and in Japan they say it is a Shinkansen. So those people who are into that program, particularly the chief who initiated that bullet train or the super fast train, was invited to kind of present how this can be formally evolved in India, how this idea of super fast trains can be done in India. As Ulrich may know, the methodology which is adopted in Germany as well as in Italy for these super fast trains. So this was the first initiative by one of the professors in a small place known as Kharagpur in the part of West Bengal, the eastern side of India. And subsequently, it was a very big success, and it came to the notice of the Ministry of Human Resource Development, and they said, we would like to have such kind of programs in other institutions of national level importance, to initiate more activities which will gain both societal and economic relevance to the progress of the country. So in this manner, a big program was formulated, and we from Anna University or from university side were invited as to how this can be taken up in universities, because it was all given to national institutions at the initial stages. And once again, I want to say to the audience here that such programs are not a fallout of a lecture program. This is a thinking which goes behind the evolution or the progress of the nation as a whole. So all of you who have been participating are one way fortunate, and in another way you are going to be the future of the national development and in the way humankind evolution or development. I should also thank my longtime friend and Professor Sendil Kumar, the Dean of the campus at Trichy. He had been one person whom I have met in my life, who was always extraordinarily kind, polite, but at the same time very active and supportive of all the initiatives of the faculty members in any academic and research activities. So to have this kind of a program in a place like Chennai, it's not very difficult, or I should say it is comparatively okay. But to organize it in Trichy in online mode needs a lot of continued efforts and involvement. So I also would like to appreciate the other colleagues, the head of the pharmaceutical technology, Dr. Tiruchirappalli, and Dr. Rukmani, who are all been supportive to people like Dr. Selvamani. Dr. Selvamani has very good experience, and he is another excellent person who always tries to keep in touch with the evolution or the progress of the university. The other thing, I went through the bio of our curriculum vitae of Professor Ulrich. I can speak in very simple Italian, but I thought for the convenience of others, I'm just trying to explain. In Italy, the methodology to evolve research programs or active academic is very different from India. In India, we have a still very structured learning, or what we call as a pedagogical learning, which means the teacher keeps talking, and then subsequently the student takes note of the discussions of the presentations, and then at odd times they make some clarifications or interactions with the faculty. Whereas in the time I spent in Italy, whatever I know of the European culture, the participation by the listener is more active. And in the sense, they keep interacting very normally, and then the professors are also very accessible to the student community. So I'm sure Dr. Ulrich had a very good time in this forum to have an active interaction from the participants. And if you have any time, I request you to kindly visit India at the latest possibility, because in this part of the country, particularly in the institution where Dr. Selvamani works, and in the present place where I work, and also at Anna University in Chennai, we have a lot of activities related to biotechnology, and we are looking forward to establishing a vibrant program at more at an international level, because the so-called people who had built these institutions in this state or in this part of the country had been very vibrant, and they have made very significant fundamental contributions. Just to say a small note on the university which I am working currently, they had got a very good recognition at the national level. This is known as a prize, or you can say it's a simple recognition, which is called as the Bhatnagar Prize, which is the highest prize or the highest recognition rather than a prize, in the name of a scientist by name Santiswara Bhatnagar. And from this university, 4 people, particularly from biotechnology related domains, have been recognized as Bhatnagar Award winners. So this has been one of the very active places, or it continues to be one of the very active places, because we also have currently many active groups working on various aspects of biotechnology, plant biosciences, molecular biology, and genomics activity. The one thing which impressed me is about the 2-point method to the present method of cancer detection, and subsequently to evaluate or stop the proliferation. This is very important for the present world, and we have done a very great effort to stop this corona or COVID-19, and therefore I hope within the next couple of months, we should be able to move a little bit better with respect to traveling from place to place, which will give exposure to the student community and to the fraternity in the teaching, to interact with experts like Pfeffer. Because the purpose of this program, as I told initially, was to evolve active research and make progress in technology-oriented activity. For example, I have heard about many times about the various tools used. One such is CRISPR, or what you call as CRISPR, which could be understood better if you are around here and if you have shown something as an hands-on to the participants. They would like to evolve this as a more serious issue or an activity of progress. And in particular, we are very active, as I said, in genomics activity. And there are some possibilities we can think about in this part of the country or the so-called state, particularly the place now I am working, Madurai. We have solid evidences of the human evolution in terms of the remains which has been identified or excavated, and then they have done a lot of analysis to show that this particular population had been in existence about 2,800 before Christ. So this is one of the very important findings which has happened in the last few months, which has been scientifically established. It's not that somebody says that we have done an excavation and then we have some samples to show that we are the oldest humankind, or the kind of thinking we can expose as a pride part of this. The other link we have with Italy very strongly, though I had been there several times. I had lived there for 6 years. I visited almost every part of Italy, and my son is in Berlin. So I have a lot of connections with the European part. The most important remark I should say is that we want to evolve a national level or international level program, and I would seek your support to interact. I will write to Professor Ulrich in this regard. The genomic activity we contemplate could make a lot of progress to say, for the participant's sake. There are a lot of human remains which has been found in this part or in this city, and this human remain has to be very carefully handled to understand the genomic character. And the group here has published in Nature to do this research and then establish a procedure to handle these human remains, so that they don't manipulate the data, they don't give a false hope on the inputs. The other extreme side of it is every nation is now spacefaring, in the sense they want to go to the moon, they want to go to different planets. So one of the idea in Indian thought process is that India has procured about a couple of kilograms of the moon's soil from NASA. And from that moon soil, they are making an artificial soil or a synthetic soil, which will be as equivalent to the moon surface. The idea is to create an igloo kind of structure for the man to go and stay for some time to do a research, or from there to go to other planets. So that moon becomes a landing point, like you go from one place to another and you have a stopover. So they want to establish this kind of thinking. But the real problem they have is whether even in that short time, whether man can exist on his own with the available resources on moon surface. So for all this, we need a lot of help from people like Selvamani, who is very active and young, to promote this kind of thoughts. And Professor Ulrich also can guide whether we can really take over such kind of initiatives, or whether it is going to be just a collective data, and this data is only for understanding and for the students to handle real-time problems, like you have some cancer and then you have some remedial procedure or a therapeutic approach, and then you try to live in a longer methodology. Beyond the longevity of humankind, we should have some thinking whether we can go into another environment and mankind can exist, because you have now such tools of genomic trimming or genetic modification, or if not modification, at least some kind of trimming to express the gene in a better way so that it can extend its stay in an outer space or in moon. So these are some of my thoughts I wanted to share. Particularly, I want to invite Professor Ulrich, whenever it's possible. We would like to have you here with us so that we can have more discussions. We can submit a joint proposal. There are various agencies which I know of now who can submit, who can support in a collective way. The last thing I would like to tell the young audience here is that the Government of India has initiated various proposal invitations, and one such is what is to be termed as glue, G-L-U-E. Glue refers to using a paste or some subject or some adhesive to join two pieces of material. Like you have a metal or you have a non-metal, and if you want to join them, now we have various forms of materials which can join this metal with a non-metal, or two broken parts of a non-metal like a plastic or a polymer can be easily joined. And even you can take two pieces of paper and then put some glue to make them join together. This kind of a concept is getting evolved, and the Government of India through DST has announced a major funding of about 5,000 crore, which in European terms will be about, if I don't make a mistake, something like 5 crore is 500,000 or close to 0.6 million euro. So you can multiply 0.6 into 1,000, which is about 600 million euro. This is really a big budget, but that budget is going to be given to groups which are going to make a collective idea, not just funding a particular project. But if you have a larger program in mind, it can be in electric vehicle, it can be in biotechnology, it can be in programs which is to do with healthcare. So they are going to fund it in a very big way for the next couple of years or close to 5 years, about 600 million euro, which is a very great number in any country's standards. And that means young minds should come forward to write a proposal with new thoughts, and the facilities are already existing in many parts of the country, or the samples will be available, as I said, from the excavations recently made, but you need to only look at those samples very carefully. And for that to happen, you may need some facility or kind of an expert like Dr. Selvamani and Dr. Ulrich. They can come forward to link these young minds with this budget offer, and then they can also bring about new ideas to practice by having new equipment if necessary, or a new facility like a standardized environment to handle samples. So all that can be made possible because Government of India is coming forward, and this is a huge budget that has already been identified in some form on the areas to work, like as I said, electric vehicle and so many other areas. And they have also identified some cities like Bangalore, Hyderabad, Pune, Calcutta, Delhi, because there are very good government laboratories already existing. So they want to provide only this kind of glue, which will kind of pick up all these facilities which are existing, the intellectual minds which are existing, bringing them together and then bring about good programs for socio-economic progress in the way of research and development. So this is another big opportunity, and I just want to bring it to the notice of the participants here, because many of the others who are here as faculty or as the team which is coordinating, you should encourage the youngsters to interact and gain experience from experts like Dr. Ulrich, so that this will be a reality in the days to come. So thanks for this opportunity. I took a little bit more time to express myself, but I hope I contributed to the wishes of Dr. Selvamani. Thank you.
Thank you so much for your words.
Thank you.
Sir. It was very nice, sir. Thank you. It was very much inspiring. Thank you. So next, I would like to call upon Professor Rukmani Madam, Director, Center for Excellence in Nanobio Translational Research, to say a few words and felicitate this program.
Thank you, Dr. Selvamani. Am I audible?
Yes, madam.
Yeah, thank you very much. Most respected Honorable Vice Chancellor, Dr. Kumar.
All the participants. And I must thank Professor Kumar here, Honorable Vice Chancellor of Madurai Kamaraj University, who is a renowned professor and highly notable researcher in Anna University, as well as an able administrator. Whenever we go with a problem, he is very much supportive enough to have a vision in this thing and to solve this problem and make sure that it will not happen in future like that. Such a visionary leader of Anna University. And it has been our fortune to find his presence here. And this distinguished presence is very much inspiring in the valedictory address today. And he has made an open invitation to you for a collaboration in future, because their biotechnology department is one of the pioneer in India who had established the historical biotechnology earlier days. And we feel as a student during my student days, they are the giants of biotechnology. So, and the invitation from the Vice Chancellor is very much making a sense of honor to me. And you can also think on it. This will be a fruitful thing for future research progress. And I must thank Professor Rukmani, a senior professor from my department, as well as the director for the Center for Excellence in Nanobio Translational Research, who consented to felicitate. And the words, always she is appreciating and us to do something. It is all based on their appreciations, and they are all a very good well-wishers to me. Thank you so much, madam. And I submit my hearty gratitude to all the dignitaries who have decorated the virtual dais today. And it's my proficient sense of gratefulness to all our guests as well as to the participants who had spent their valuable time, patient listening, interaction during the course, as well as following all the instructions and norms of the GIAN. So you are very much cooperative. Thank you, audience. And I must thank all the administrators, directors, heads of various departments, faculty colleagues for their encouragement. And Professor Bhaskar from my Vice Chancellor, Madurai, Manonmaniam Sundarnar University, and Professor Subramanian from NIPER, Professor Suresh Kumar from VIT, Professor Prem Kumar from Bharadasan University, Dr. Ravi Varala from Rajiv Gandhi University, Dr. Alam from King Khalid University, Saudi Arabia, and Vijay Kotra from Malaysia for all their support by way of participating in this course. And I must thank the alumni from my lab, Dr. Arpuda Bibiana, Dr. Sendil Kumar, Dr. Anish Kumar, Mr. Krishnanand, Mr. Satish Kumar, and all other members of my lab and student volunteers from the department who handled the event throughout in various aspects. And not the least, I sincerely thank all the participants, as well as there are many online viewers. There is a good statistics. All your lectures had been downloaded and viewed several times in the Facebook as well as YouTube, which was live streamed during the course of lecture. And I must thank Mr. Babu and Director U&I Media and Mr. Shankar, liveibc.com for technically coordinating this event. And their service is wonderful, because I need not worry on these aspects. So they are doing a wonderful job. And I must thank Ministry of Human Resource Development, Government of India for the generous support under the GIAN scheme, without which this course is not at all possible, that enabled advanced learning in India. And I strongly believe this course will be a new beginning for new collaborations and to initiate tumor biology translational research in India with the support of San Martino, Italy, and other institutes in Italy. And thank you, Chief Guest Sir, Professor Kumar. Thank you, Professor Ulrich, and thank you, Dean Sir. Thank you, HOD Madam, and thank you, Director, and thank you all the audience. Thank you, one and all. Thank you.
Thank you.
So we are coming to the end. So there are few announcements. So there is a evaluation exam in the Google Classroom scheduled at 8 p.m. to 9 p.m. today. And please don't forget to submit your feedback in the GIAN portal available in the participants login today. So let us all stand for the national anthem. Thank you.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जन गण मन.
Thank you so much.
Thank you, Selvamani. Thank you.
Thank you.
Yeah.
Thank you so much.
Congratulations, Selvamani.